Tracy Castellano and
Anna
Dial. So 2 for the price of 1 here, folks. You get to ask lots of extra questions, I hope. Both are with FWC, and the title of their talk this afternoon is Effects of Experimental Translocation on Gopher Frog Survival and Movement.
So Tracy and I did a study on gopher frog translocation and experimental translocation, and we also have several co-authors because, as you'll see later on in the talk, we pulled in data from 2 previous studies to analyze along with our data. One of those was done by Anna Farmer, and then we have Javin Botter, who helped with our analyses, and then the 3rd study that we pulled in data from was by Betsy Rosnick and Steve Johnson. So why did we set out to study gopher frog translocation? Well, for starters, it's a species that's declining in parts of its range and federally petitioned for listing as threatened. It's a high priority gopher tortoise commensal species. Until 2012, FWC policy allowed translocation of tortoise commensal species along with tortoises. But then in 2012, concerns about impacts to commensal populations led to a halt in translocation pending research. So basically a realization that the background research had not been done on the commensals to see were there potentially negative effects happening with the animals being moved and also with the animals in the already resident populations. So concerns like competition and disease and survival of the translocated animals. And then lastly, there are few studies on translocation of amphibians and no published studies on adult gopher frogs. And then one more reason, we have a species action plan that FWC has written for the gopher frog, which you can find online if you wish to look for it. And this has specific conservation actions written out for the species. And one of those was to get at this question of informing FWC policy about whether or not commensals could be translocated in the future by doing a radiotelemetry study on the gopher frog and seeing the effects on survival, movement, and behavior. So what are the potential risks? I already alluded to this somewhat. One is that amphibians have strong homing mechanisms, so they may try to return home upon translocation. Many species, including the gopher frog, are able to move between uplands and then the breeding ponds. Numerous translocations of amphibians have failed because animals have dispersed upon release, and that can often result in death. Translocation may negatively impact the recipient population, and death of translocated animals could happen through a variety of ways. Predation. If there are roads nearby, it could happen through road mortality as well. So we conducted a pilot study in experimental translocation at a single site in Ocala National Forest from 2015 to 2017. Our objectives were to evaluate effects of translocation on survival and movement, determine if translocated individuals are able to successfully navigate in a new environment, and evaluate feasibility and methods for a larger study involving multiple sites. We had 6 capture sites. We captured frogs both at burrows and breeding ponds, 1 isolated release site with a single breeding pond, and the capture and release sites were greater than or equal to 5 kilometers apart to try and minimize any homing behavior. The captured frogs were immediately translocated. They were released into tortoise burrows, and then we used radiotelemetry to track them for greater than or equal to 30 days, and then less often until the transmitters failed. And we actually went through a few different iterations of methods for this study, which we don't have time to get into, but it did take a bit of work to figure out what methods worked best. So for our comparison groups for analyses, we had non-translocated frogs from this study and 2 previous monitoring studies, which I mentioned with our co-authors. So we had 6 frogs, non-translocated frogs, we looked at from our capture sites in Ocala National Forest from 2015. There were 7 that we looked at from Anna Farmer's study in Jennings State Forest in 2014, and then 11 from the previous study at Ocala National Forest in 2007 by Betsy Rosnick and Steve Johnson.
Okay, I'm going to talk about the analyses and the results. Can y'all hear me? Okay. So we used a survival analysis, and then we also looked at movement metrics. The most important metric is daily probability of movement, because like we alluded to earlier on, movement was the response we expected, and it turned out to be a very important factor. So daily probability of movement, although we monitored our frogs for the most part daily or every other day, because we were using data from other studies, there were some where they weren't monitored quite as often. And so this allowed us to predict the probability of movement for days when we didn't actually monitor the tortoises, the frogs. Oh my gosh, the tortoises. So daily probability of the movement is just the probability that they move from the spot that they were to a new spot on the next day. We also looked at days spent at the surface, which is, by surface we mean outside of a burrow or other type of refugia. The numbers of distinct locations, so some frogs might have moved a lot, but they moved back and forth between maybe 2 locations, whereas others moved across the landscape and used distinct locations. And then their maximum displacement distances. And we also looked at homing behavior, so orientation angles back toward their release sites. So the results. We ended up monitoring 23 translocated frogs and 24 non-translocated frogs. The duration of monitoring was approximately the same, but some of them were monitored up to about 5 months. So there was a lot of variance in the duration of monitoring. We had 7 known deaths among the translocated frogs and 4 known deaths among the non-translocated. And I'm giving you this percentage here, the apparent survival rate of 67% for the translocated versus 80% for the non-translocated. But I want you to, people want to know this number, that's what people ask, but I want you to understand that this is not, that is very rough and dirty. There's a lot of sources of potential bias because they were monitored for different lengths of time. We excluded the ones that were lost almost immediately in less than 30 days, but then some of them were lost after we had already monitored them for months. We didn't want to throw those out. So if these are biased, they're biased high. The survival rate was probably a bit lower than what we were able to document. An important point is that the deaths for the translocated frogs occurred almost immediately, whereas deaths for the non-translocated frogs happened later on. And the causes of deaths were predation and 1 roadkill. So our survival analysis was very informative. I'm going to try not to get too much into the weeds on this one. There was a very strong negative effect of probability of movement on survival for both groups, for the non-translocated and the translocated groups. The survival increased after the first month. That wasn't a significant effect, but it did increase after the first month. So the dark lines represent the first month of data, and then the lighter lines represent later months. I think that the reason for that is that the non-translocated frogs were moving around in ponds. Ponds are dangerous places. There's a lot of predators at ponds. For the translocated frogs, I think the reason is because they were learning to navigate in a new environment. Once they knew their way around a little bit, they could move back and forth, maybe without getting killed. There was no effect of translocation on survival after accounting for daily probability of movement, but that doesn't mean there was no effect there was a significant effect of translocation on probability of movement. So the principal thing that caused death was movement, but there was more movement for the translocated group. Does that make sense? Okay. But the effect wasn't huge. Daily probability of movement was higher, significantly higher in the first month than any other month for both groups. It decreased over time, but it stayed a little bit higher for the translocated group out to about 5 months. Although when we're getting toward the end here, we're monitoring very few frogs. So there were essentially two groups within the translocated group, there were essentially two groups. There were about half of the frogs dispersed almost immediately within 2 weeks, and they almost all died. There were about half that didn't disperse and stayed relatively still, and they mostly lived. So the probability of movement was significantly higher for the ones that died. So these two groups are about roughly the same number of frogs. This is the probability of movement for the ones that died. This is the probability of movement for the ones that survived. So if they survived, they stayed really still. We were surprised that the non-translocated frogs had higher movement rates in the first month. We assumed that the non-translocated frogs were going to be still and that the translocated frogs would move, until we realized that about half of our frogs were caught at breeding ponds. So the frogs aren't necessarily moving more when they're visiting ponds. They're moving around within the basin of the pond while they're at the pond, and then they leave and they move up into the upland. So we're tracking that movement. Since we caught them at the ponds, then that's the first month of monitoring. So we looked back at the data, and sure enough, there was a significant effect of having visited a pond. Daily probability of movement was significantly higher for frogs that visited a pond versus the ones that didn't. So this figure, just for illustrative purposes, shows the movement paths of the translocated frogs. This was our little breeding pond, and we tried to release them pretty close to the breeding pond, as close as we could, where we could find burrows. And then that shows their movement paths, and the circles are the ones that didn't move at all. They just stayed in their release burrow. So irrespective of translocation, the frogs that died, there was no significant effect of translocation or not, but the ones that died spent more days at the surface, so they're moving around outside of refugia. They used more distinct locations, and these were marginally significant. Those weren't significant effects. And there was no evidence of homing that we were able to detect. There was no evidence of long-distance dispersal, although keep in mind that we lost some frogs, and that might have been because they were dispersing. And there was no evidence of movement back toward the capture site. So in summary, what we found out is that movement is extremely dangerous for gopher frogs, whether they're translocated or not. Translocation does appear to stimulate high rates of movement, but those rates of movement decrease over time. Our survival curves showed us that frogs that don't move have a very high survival rate, approximately 90%. And frogs that have been translocated and live long enough to settle down have a high survival rate. So we saw evidence of directly 33% mortality. It might have been a little bit higher. That is high. It could have been much higher. We wouldn't have been that surprised if they had all died. But if they had stayed at the site they were taken from, or if it were a mitigation, not in this case because it was experimental, but if it was a mitigation translocation, then if they stayed at their capture sites, there would have been 100% mortality. So our first question was, is there a benefit to the translocated tortoises? I did it again. The translocated frogs. I think that the answer is yes, some of them can be expected to survive. The question that we still need to answer is, first of all, can that survival rate be improved? And second, does that benefit outweigh the potential risks to the recipient populations? We also need to know if there's long-term survival, if the ones that we release stay there, and if they breed and contribute to the population growth. So to assess that, we're taking genetic samples from tadpoles. We took a sample from each of the frogs that we released. So we're going to look for a genetic signal that they stayed in place and contributed to the population. There still needs to be a lot more research. This needs to be replicated at other sites. We need to assess disease risk and other consequences for the recipient population. This particular study did not attempt to do that, and that's an important question that needs to be addressed. And I think that it would be reasonable at this point to assess soft releases. We know that the principal cause of death was immediate dispersal from the translocation site. There have been soft releases used for geckos, and it was successful, so I think it might be worth testing on frogs. And we want to acknowledge our funders. Deborah Burr and the Gopher Tortoise program at FWC funded most of this project. We had a lot of support from other people. In particular, I would like to point out Matt Fredler, the frog whisperer. He caught essentially all of the frogs for our last season when we weren't able to catch any. And Gabriel Camner, he's working at Archbold now. I cannot overstate how helpful he was. He provided us about 5 frogs from his own study site, and then he also volunteered with the day-to-day work on our project, and we're very, very grateful to him. So, any questions?
Sorry.